Thank you for the opportunity to speak. I've been at Harvard for 32 years, but for the last 7, I've been the founding director of the Wyss Institute for Biologically Inspired Engineering. And great, I think we now got slides. So just this is basically our logo. It's one of self-assembly. We started with basically me, and we hit 375 people in about 3 years, which is where we plateaued. Just one short note on the backstory. Essentially, the vision of the Institute is that engineering has transformed medicine over the past 50 years by taking engineering principles, trying to solve medical problems, hip implants, pacemakers, drug delivery, et cetera. We feel that we've uncovered enough about how nature builds, controls, and manufactures from the nanoscale up that we can now leverage biological principles to develop new engineering innovations. So that's what we call biologically inspired engineering as opposed to bioengineering or biomedical engineering. We got kickstarted with the largest gift in Harvard's history of exactly $125 million. And we were tasked with taking on really high impact, high value problems. And the biggest problem that I could see at the time is that the drug development model is broken. You talk to CEOs, which I have done, of major pharmaceutical companies and biotech, and they say the big challenge is to fail quickly and cheaply, which is so depressing to me. And why is that? Well, the study at Tufts says it costs $2.6 billion to take a drug through full approval. There are many reasons for failure, but a lot of the preclinical early studies use cells cultured in dishes. They don't function anything like our bodies. Animal studies take years to complete. Ethical issues are huge, especially in Europe already. Will spread here soon, I predict. But the real problem is that more often than not, the results do not predict what happens when they get to human clinical trials. It's like telling a grad student to define your next experiment based on your failures instead of successes, but this is the way the industry works. And as a result, there's a lack of drugs reaching patients. So many people have searched for models that would mimic whole organ function. So when we started the Wyss Institute, we have 6 platforms, and one I formed and I lead called Biomimetic Microsystems, and the idea is to engineer microchips containing human cells that reconstitute organ-level functions, not cellular tissue, but organ-level functions for drug screening, diagnostic, and therapeutic applications, and to sort of solve this problem. And it's a collaboration with many faculty, including George Whitesides, who's here. Now, why microchips? Well, microchip manufacturing offers control of the features at the same size scale, nanometer to micrometer, that living cells and tissues live at. And George and I started over 20 years ago, I think 25 years ago at this point, to take techniques from microchip manufacturing, which George innovated, soft lithography, microcontact printing technique, and we used to control cell adhesion, and we showed we can control cell fates based on cell distortion and mechanical signals. And George started to develop microfluidics, and we collaborated together working with some cells cultured in them. And many of you have heard of what the power of microfluidics and miniaturization of instrumentation. But to me, as a cell biologist and MD by training, these are microvascular networks. And so we started to think of how we could combine all this to really mimic organ-level structure and function. And we had a first breakthrough published in 2010 that in Science we called a human breathing lung on a chip. And the idea was that one of the major functional units of the lung is the air sac or the alveolus. It's actually a relatively simple structure. If you go to EM level, there's air, a single layer of epithelial cell, the alveolar cell, a flexible porous extracellular matrix, the basement membrane, and on the opposite side of the same basement membrane is a single layer of capillary endothelial cell, and then there's blood. But the thing that's not shown here is that this is a mechanically active structure and that we breathe in, we breathe out, blood is flowing, and the cyclic distortion we know from respiratory physiology is absolutely critical for regulating development and function, including gene expressions like surfactant and many other functionalities. So if we're not trying to engineer an organ, we're trying to distill it down to its minimal design features so that we could recapitulate organ-level physiological function. So if you distill it down, what makes an organ? It's 2 or more different tissues that interface and new functions emerge, often involving a vascular tissue, a blood vessel. In our case, we also thought that flow and breathing motions were important. So I'm going to show you an animation that shows you how this works. At the top right is a picture of the chip. It's optically clear. It's made out of silicon rubber. It's the size of a computer memory stick. If you cut it in cross-section, it's got 3 parallel microchannels. The middle channel is spanned horizontally by also silicon rubber, very thin, with engineered holes about 5 to 7 microns, coated with extracellular matrix. We then plate human lung alveolar cells on top, human lung capillary cells on the bottom. We just recreated the alveolar interface. The trick is that we then put cyclic suction through the side channels, and because it's all flexible rubber, it actually stretches and relaxes the same degree of strain or distortion when you're breathing in and out this minute. We then can flow air in to create an air-liquid interface, and we flow medium with or without primary human immune cells, and as you'll see later, we could do whole blood. So if this works, we should be able to mimic organ-level functions. Let's say in an infection, you have bacteria. That actually activates the epithelium to secrete cytokines, which then activate the endothelium to express adhesion receptors that pull circulating immune cells out of the circulation. They adhere, roll, diapedese, go through, and engulf. So what I'm going to show you now is high-resolution imaging in the device, and we can do any imaging you can do in culture. These are primary human neutrophils. I know they're fresh because we took them out of my postdoc, and as you can see, the endothelium you can't see labeled as quiescent because they just flow by, and the epithelium is on the other side of the screen. We now put a bacteria on the other side, and now they're activated. They express ICAM, among other adhesion receptors, and the white cells stick. Now you can go to higher resolution imaging, as you'll see in a second. The holes engineered in the membrane are pentagons. White blood cells migrating on the endothelium. About there, it finds a space, goes through, finds the hole in the membrane, wiggles its rear end through the matrix, goes out of focus. You can see it come out by phase contrast. And I'm now going to show you the white blood cells red because I'm going to show you the GFP labeled bacteria green. And you see them being engulfed. So you just watched the entire all human inflammatory response in real time at high resolution in this little rubber chip. So I was funded, actually George and I, it's a long story about this grant, but it's the only grant I think we ever got in nanotechnology, was on nanotoxicology. And it's amazing as a cell biologist that particles 100 nanometers or more get across the blood-brain barrier, seminiferous tubules, never really made sense to me. But many of these enter through the lung, especially airborne particulates and smog. So we actually carried out studies where we used what researchers in the field use as simulants for smog particulates. They're 12 nanometer silica particles. We could do microfluorimetry, so we measured oxygen-free radicals in the cells. And what you see here is quite interesting. If you have the cells under static conditions, and you give them with or without these pollutants or these nanoparticles, there's no injury response. If you give them breathing motions, physiological breathing, there's no injury response. But if you have physiological breathing and these nanoparticles, you activate reactive oxygen species and you recruit circulating immune cells and mimic inflammation. This would have never been seen in a static culture, transwell, et cetera. Furthermore, if we can measure absorption and bioavailability, if you give the nanoparticles to the airspace, you can measure what goes across the 2 cell layers in the matrix, and I don't know if it's dark enough, but basically we saw an 8 to 10-fold increase in the absorption of these nanoparticles across this. There was no change in absorption of fluorescent albumin or dextran. This is not junctions opening. And it was mechanically induced, so we think it's actually a mechanically induced transcytosis response across the 2 cell layers, where it goes through the cells, spit out, through the matrix, taken up by the endos, through the cells, spit out, and never really been seen before. Now, this isn't mimicking physiology. This is a prediction. No one's ever seen this before. So we developed an ex vivo model of ventilation perfusion in the mouse where we could use a nebulizer, give the same nanoparticles. We could control whether or not it breathes, and we got the exact same sort of response. And this was a full article in Science many years ago, and we've been told that this sort of changed the paradigm, certainly the funding pipeline, that this is possible. Now we started to talk to pharmaceutical companies and they said, this is really interesting, but the real problem is disease models. That's where the animal models really fail. So we took this, and also there was obviously interest in toxicology. So we took this and we basically hit 2 birds with 1 stone by making a model of drug toxicity and also a disease model. We used interleukin-2, which is an approved cancer drug, and its dose-limiting toxicity is pulmonary vascular leakage causing pulmonary edema, fluid on the lungs. So we injected this drug intravenously like in a patient. And what we saw is it's time 0 with air above the epithelium. This is what it looks like. At 2 days, you see a meniscus filling the upper channel. And by 3 to 4 days, it completely fills with fluid. That is the exact same time course with the exact same drug that this side effect is seen in humans. We quantitate this. We use a trick from kidney physiology. We use fluorescent inulin. And what you see here is the red is a static chip. This little increase is what interleukin-2 does. But this is with physiological breathing motions. Again, we went to the animal model. We saw the same thing. Now, this is important clinically because most patients with pulmonary edema are on a respirator, a ventilator, so you could actually play with mechanical ventilation, perhaps have some effects. But I've worked on mechanobiology for 40 years, the idea that mechanical signals are as important as chemicals and genes for regulating cell fate and physiology. And I had shown that the fastest mechanical signaling that we'd seen in endothelial cells when you stretch a cell is 5 milliseconds, and it's activated through forces going to integrins, going to the focal adhesion, going to a stretch-activated ion channel called TRPV4. And I heard that GlaxoSmithKline was working on TRPV4 inhibitors. And they didn't fund the work, but they gave us the drug. And we tested it, and we completely knocked out pulmonary edema in this model. Now, it turns out that they were kind of shutting down that program, but they then did this study in dogs and rabbits, I think it was, with cardiogenic pulmonary edema and got the same results. And we actually had back-to-back papers in Science Translational Medicine. And that drug, I believe, is in phase 1 clinical trials. So this one chip basically provides proof of principle for drug efficacy, drug toxicity, and human disease model. Now, the companies that came to us, the pharmaceutical companies, they said, very exciting, but the big problems we're facing are asthma and COPD, and they're really largely inflammatory diseases, as are many diseases. So that's a disease of the small airway, the bronchiole, not the alveolus. So we took our chip, we made the airway space 1 millimeter high, which is the radius of a small bronchiole. We got primary human alveolar epithelial cells, and we cultured them on it. And this is in vivo. They grow as a pseudostratified mucociliated epithelium. And when we put them on the chip at an air-liquid interface for 3 weeks, with being fed through the capillary side, just like in vivo, this is the chip, and the top is the epithelium, pseudostratified, and the endothelium in red is at the bottom. If you now stain these for cilia in white and mucus in magenta, you can see that it's a ciliated epithelium across the entire chip, millimeter chip. And if you go to the EM level, this is what it looks like. Pathologists said this was like a human airway, but this is the chip. And these actually move, and they move in a directed way. And what I'm going to show you next is if you give a fluorescent microparticle to measure mucociliary clearance, this is what you see, and this is real time. So this is mucus clearing on the chip at the same rate exactly as we are clearing, or you are clearing as you're listening to me. So basically, this is a little human airway on a chip. The ciliated cells, goblet cells, club cells, basal cells are in the same percentages. The cilia have the same structure, function, et cetera. So we started to explore disease modeling, and we didn't have approval for viruses yet, so we used a viral mimic, poly-IC. And what I want to show you here is that you get cytokines, and this is IP10, Rantis, and IL-6. But what you see here is if you have a tissue on a chip in this gray bar, just the epithelium, versus the endothelium on a chip alone, which is the second small bar, and both together when you have an organ on a chip, there is synergy in terms of the response that you would never see normally with either cell alone. And this is known to happen in vivo as well. We've also started to do viral infection. This is H1N1, influenza, you could see. But more recently, we're starting to explore how this sort of platform might be used. And what I'm going to show you here is Sendai virus, GFPs. So you're going to watch infection in real time on the chip while measuring cytokines that are flowing out of the effluent, because we could collect both the airspace channel if we have fluid flow, but usually the vascular channel, which is what's here. So now I'm going to click it, and you're going to see it infects. Before you see anything, the IL-6 goes up and while it's infecting. And then it's later that Rantis comes up, And we have microelectrical sensors. We can do 64 different things now, anything you have an antibody for, basically, in nanoliter volumes that come out of this. So you could start watching human responses in real time. In December, we had a paper where it published in Nature, I don't remember anymore, Nature Methods or Communications, that we took cells from human patients with COPD. We made the same chips, they looked the same, but after 3 weeks of differentiation, they retained the phenotype of suppression of toll receptors that COPD patients are known to show in vivo. And if we give them simulants of either viral infection, poly-IC, or bacterial infection, LPS, endotoxin, we could see that the COPD patients have enhanced sensitivity in terms of cytokine production of IL-8 and MCSF, whereas there's no change in IP10 and RANTES. So we're mimicking exacerbation of COPD on chip, which has never been done before in vitro. And I should say we identified a new biomarker out of that. Now this is unpublished. We can now have a smoking chip. This is like a Gatling gun of cigarettes. It has an automatic lighter. They actually smoke. You can control the puff intervals, and it goes right into the airspace of the chip. It's really kind of scary, actually. But we don't do it in the lab, only in the hallways, basically, in the stairwells. So if you put these cells, COPD chips versus healthy, you could actually see exacerbated responses of COPD patients relative control, cytotoxicity, cell stress, cytokine production. And this is gene microarray of our chips at the right, and past publication of human smokers with COPD, and they are very, very similar. And we've identified some new targets, we think, potential therapeutic targets. So we've also done other organs. So this is, we call the peristaltic human gut on a chip. We took the same chip, we made it higher, wider. We now gave it peristaltic-like deformations, trickling flow like in the gut. We started with an established cell line, human CACO2 cells, that actually came from an intestinal tumor originally, but have been used by pharmaceutical companies for years as a barrier test. It takes them 3 weeks on a transwell to get a barrier, and they look like squamous epithelial cells instead of columnar cells like they do in vivo. In 3 days, with peristalsis and trickling flow, this is what our chips look like. They form villi. They spontaneously form villi. And if you know the intestinal villus, it's a very unique structure. It has proliferating stem cells in the crypt, at the base. They then differentiate into 4 lineages in small intestine as they migrate up the villus. This is a 2-hour pulse for proliferating cells on our chip. They're in the crypt. You wash them out, they go up. They differentiate into the absorptive, goblet, endoendocrine, and Panda cells of the small intestine. This is barrier function. This is 3 weeks the way pharma does it. This is 3 days on our chip. This is pericellular permeability. This is a specific differentiated activity. This is P450, which is very important for pharmaceutical companies. The gut metabolizes drugs. No activity in regular culture. This is on chip. And this is mucus production. Now, this is important because these cells, because they're differentiated and there's flow and they put out mucus, we could do what I think is one of the most important things we've done. We could culture live microbiome for weeks in a symbiotic relationship. Most work on microbiome, which is the major paradigm shift in medicine in the last 10 years in my mind, is genomics. But very few people can study the interactions with human cells. So we started, we started with Lactobacillus GG. It's a probiotic originally isolated from human gut. Find it in yogurt. You put it in a transwell. In 1 day, you have contamination. You lose barrier function. Co-culture it for weeks. You get better barrier function, just like people claim probiotics do in vivo. More recently, we've done 8 different microbes. They're squiggling here in this movie. They live in the crypts very happily, symbiotic relationship. But if you now do gene microarrays on these, and this is in duplicate, and you compare transwell to the, if you compare the transwell to the gut chip, they're totally different across 22,000 genes. If you now just add these gut microbiome, these probiotics, they're totally different again based on gene microarrays. And they're getting closer and closer to the terminal ileum, which is the part of the small intestine where you actually see microbes. So I say there are no bad cells, they're just bad microenvironments, just like there are no bad kids. You know, if your kid goes bad, maybe you bring them home and they'll come back. We seem to be able to do that with cells. So if you put pathogenic enteroinvasive E. coli, now they overgrow. And we recently published in PNAS a model of inflammatory bowel disease where we could look at the chip alone, and this is the villi from the side in white. If you put enteroinvasive E. coli, you basically lose the villi like you do in patients, and you lose barrier function, as shown here. If you give a lab strain, you don't see it. If you give endotoxin, you don't see it. But if you now add human peripheral blood mononuclear cells and you watch the contribution between the two, now you can get complete injury. But because we could measure effluent in real time, we could actually measure cytokines, and there are no cytokines secreted apically. They're all through the vascular channel. And only when you get injury do you get 4 cytokines. And we looked at 12 in the literature implicated in this disease. And that combination of 4, if you give any one alone, you don't get it, 2 alone, 3 alone, but all 4, you get the same exact phenotype. And we can give it at the concentrations we measure in the chip. So we know it's relevant here. So I think we're going to begin to identify combinatorial targets. And finally, if you stop peristalsis, you get bacterial overgrowth. And this is why surgeons always want to get your gut moving after surgery and anesthesia, because you can get ileus. Well, they think it's because the fluid stops flowing, but we have the fluid flowing. It's just the mechanical deformation. So this is a new insight as well. And I just published last week sort of a synopsis of this, but the idea is that we can begin to build up complexity. It's synthetic biology at the organ level. You could start with one cell, and if you don't get the physiological mimicry, you add another, and then another, and then another, and so forth. Now, we've done many different chips. Just a quick example, this is a proximal tubule on a chip. Very simple, we just took primary proximal tubular epithelial cells, gave it flow, and now you basically see that with flow you get increased cell height, increased sodium potassium ATPase, increased aquaporin, and every cell has a primary cilium in the center, just like they do in vivo. Here you can see it from the side. You get increased reabsorption, and we showed that you could see toxicities in these chips of cisplatin that were never seen in animal models, but seen in humans, we could see them in these chips. And then finally, we just developed a human glomerulus on a chip, and we couldn't do this in the past because there were no human podocytes, but we developed our own method to create them from iPS cells with 90% efficiency. And we put them on this chip on one side, and we put human glomerular microvascular endothelium on the other. And what you see is with or without cyclic strain, which the glomerulus is known to deform with each blood pulse. And when you have increased strain, you increase the contacts between the podocytes and the endothelium, which is what happens, how the glomerulus forms in vivo. And we could recreate glomerular filtration on these chips as well. And then finally, January, we published a human blood-brain barrier on the chip. We used a different technique. We used viscous fingering, developed by David Beebe, where we have a collagen gel going through the channel and a liquid medium, and the liquid fingers through while it's gelling, and you could put cells in the collagen gel. And so you basically have a hollow tube, which you then can plate pericytes on. You have astrocytes in the gel, and you could put endothelial cells on the pericytes. And here you can see, let's see if this works. This is the endothelium in this tube. These are the pericytes. And these are the astrocytes. So we recreate this complex structure in vitro. And when we look at neuroinflammation, we find that, again, various cytokines, if you have the same cells in a transwall under static conditions, there's very little synergy between these cells in terms of cytokine production, but where you see great synergy, much more like in vivo, under the dynamic fluid flow conditions. And the last thing I'll show you is unpublished, and I think is amazing. We've covered the bottom channel on all 4 sides with endothelium now. And now you could flow whole human blood without anticoagulants through this for at least 20 minutes. And if you look at thrombosis in this, if you don't have an endothelium, you get the usual platelets sticking to the collagen and just glumping down. If you have the endothelium, there's no clotting, as I just showed you. But if you have endothelium and you have an inflammatory signal like TNF-alpha, you see a very interesting response, which is the platelets stick, but they kind of stick and roll and advance along this. And if you visualize this dynamically, so that the yellow is regions of high dynamics and the blue are low, it looks like a teardrop. And this is exactly what people have seen in vivo, never seen in vitro. And this is from work with the Fury Lab at Beth Israel Deaconess. And so we're actually mimicking physiological hemostasis coagulation. So we added the epithelial layer to this. And so now we have pulmonary thrombosis on a chip. And it's interesting, it's known that endotoxin induces pulmonary thrombosis from animal studies. But when we add it just to the endothelial side, you don't get thrombosis. When you add it to the epithelial side, you get thrombosis on the endothelial side because you're inducing a cytokine cascade that activates the endothelium, and then you get coagulation. So you can dissect these processes by taking one cell at a time and looking at different combinations. Now, I formed a company called Emulate, and they published publicized that Janssen Pharmaceuticals is working with them. Janssen had a drug, a human antibody that made it through all preclinical and failed in human clinical trials because of pulmonary thrombosis. We could actually mimic that in the chip. That gets the attention of companies. So to end, I think we're up to 12 to 15 different human organs. Kit Parker's developing muscles and heart. But when I first proposed this, I suggested that because we have a vascular channel, we could potentially link all these chips by the vascular flow and create what I called an integrated human body on chips, where you could imagine that you could have an aerosol-based drug to the lung, watch it be metabolized by the liver, peed out by the kidney. Do you have heart toxicity? What does it do to bone? Or an oral drug or the same. And we actually got a major grant from DARPA to do exactly this, which was to build an automated instrument that you could think of like a multi-DVD player with each DVD holding a chip. And the idea is you could have 10 lungs, or you could have lung, liver, kidney, heart, or heart, liver, bone marrow, and that you could use computational modeling to do pharmacokinetic, pharmacodynamic modeling on chips to predict what would happen in humans. And we actually, we have now done, last year we did 4 chips for 2 weeks linked. As we are right now doing 8 human different chips linked for 3 weeks. We have to do 10 by next year. But based on the success of that, we did form this company, Emulate. I think the real excitement of this is, I think what will revolutionize the pharmaceutical industry is right now every big company spends $2.6 billion, 10 to 15 years, usually fails in a major clinical trial. They then do statistical number crunching, search for a genetic subgroup that maybe responded. Then they do a small trial 3 years later. If they're lucky, they're approved for a narrow application. With this, you could actually develop chips for subpopulations. You could also do it for individuals, but that'd be a lot more expensive. But you could do it for subpopulations, and you can imagine a Hispanic women, asthmatics who are hypersensitive to smoke, develop drugs for them, and use those very patients as your clinical trial group. And I think that is going to shortcut things, increase likelihood of success, and be transformative. That said, one of the investors in Emulate is Cedars-Sinai Medical Center in Beverly Hills, California. They're doing whole gene sequencing and IPS on every cell that comes into the hospital. And they want to use these to basically make your Crohn's disease gut on a chip or your asthmatic lung on a chip to do individualized drug efficacy and toxicity testing. And if you have the funds, I think that's really exciting too. So Harvard makes me do this. This means you don't believe anything I just told you, but most of it was done before the company formed. The company is hopefully going to sell chips and automated instruments, just small instruments that fit in incubators within the year for academic use as well as industrial. So if you're interested, keep your eye out on that. And with that, I would just say that to do this type of work requires an incredibly diverse group of skills and people, which is what the Wyss Institute Environment allows us to do. And I invite you to our website. If you're interested, we won the Webby a few years ago, which is the Academy Award of the Internet. We beat out Wired Magazine and the NASA Space Propulsion Lab. And we're bored with it now and about to change it. So before we change it, please take a look. Thank you so much.
In terms of the first part of your talk, talking about epithelial cells in the alveoli, have you determined whether those cells are making surfactant and whether those cells are type 1 or type 2 cells, and what are the ratios of each one of those?
So in our first paper, we actually used an established cell line again that originally came from a tumor, and they did produce surfactant, and the physiological breathing motions increased surfactant. Now we actually are doing type 1 and type 2, and I can't tell you the exact ratio, but we're basically getting similar results, but we're doing all normal cells. And the gut, now we're doing biopsy or organoid-based primary human gut cells, which we can now transfer to the chip, and we're getting beautiful gut on a chip with villi again. So we're moving to all normal or IPS.
I have a question about the halogen gel. Is it kind of like a hemocytometer where you add your reagent in after the cells have been growing? And also, if you're doing like T cell differentiation, if you're doing like a Th2 or Th17 polarization, like do you have to create the samples first and then add it to the
So I'm not sure exactly what you're asking, but you basically have, it's microfluidics. You have little inlets and outlets.
Mm-hmm.
You're flowing fluid through, and you could add whatever you want to that.
Okay, so can you do like cytokine expression or?
Yeah, that's what I was showing you in
stimulation?
real time, is with
Yeah, but can you
cytokine.
do polarization? Do you have to do that first? Because that takes several.
By polarization, you-
Polarization, like a Th2 differentiation. or
Okay,
a
so
Th17
we haven't
differentiation.
looked at the T cell differentiation here. We've integrated dendritic cells into the gut. We formed M cells in the gut, and we're just beginning to get to T cells and to do a lymph node type chip. But the idea is that we'd actually activate them there because the endothelium is a key player in that. And there's evidence to suggest the microenvironment is important. We also have a bone marrow chip we published that was kind of amazing, but it's a little different technology.